For our next talk now, I would like to invite Professor Marcos Quiles.
Let's start again. Hello, my name is Marcos. I'm associate professor at Federal University of São Paulo, UNIFESP, São José dos Campos, and also researcher in the project P20, the machine learning project. In this presentation, I'll give a brief overview of our development so far, our internal developments in our group, and also some developments in collaboration with the group of Professor Juarez. First, before starting talking about our developments, I'd like to talk about our mission in CINE. From my perspective, I think our primary purpose is to help to solve CINE problems via machine learning. I mean, we want to solve some problems that are undertaken by computer simulations or DFT calculations, and maybe replace some of these computations using surrogate models via machine learning, build predictors, which are much cheaper than the computations that are normally compute using a theory like DFT and so on. We also can provide some unsupervised learning using clustering and prototype selections, feature engineering, feature learning, and maybe provide some force field methods via machine learning too. And later, we aim to develop also some research in material discovery, also using machine learning. These first two talks, we are already providing some results. We have some results in these two lines, and these three, in blue and red, we still want to develop in the future steps. And also, actually, we want to help the CINE project with machine learning, but we also want to develop new machine learning methods in order to publish paper in our field. So, but I believe that this will become a consequence of the first step. By providing solutions, machine learning solutions to your problems, I believe that you are going to develop or at least improve some methods that are already published in the machine learning literature. I divided our undergoing research in three lines. One is property prediction. Given a molecule, we take some features of these molecules and compute the prediction, the properties of this molecule using, for instance, neural networks. This work is mainly undertaken by my master's student, Gabriel. We also have performed it's a mix between unsupervised and supervised learning using a graph-based approach to perform clustering. And I'll talk a little bit more about this one. And I also have my undergrad student, Philippe, which is performing, which I call biased prototype selection. It's an unsupervised learning problem. I'm not going to give many details about this research because we have the poster session. I do not want to spoil my student presentations, but what is the property prediction problem we have here? Starting from a public data set, the QM9 data set, which is a data set composed of more than 130 kilomolecules from different size. From this data set, we want to predict some properties of these molecules. The first step consists of cleaning the data and removing some anomalous molecules or molecules that have some reading problems and so on. And then we perform the second step is a feature extraction. We use the, what's the name of the package? Modred package, which from the SMILE representation provide us more than 1,800 features that can be used to feed our machine learning model. Next, we have the preprocessing, which is a normalization procedure and also elimination of some missing values or properties that have zero variance that will not help the machine learning to build the relation between feature and properties. We also apply several feature selection methods in order to reduce the number of features we have from this package and use only the most important or the best features to predict some properties. And as a predictor, we can apply several methods, but here we are using multi-layer perceptor networks. It's a universal machine learning predictor. We got very good results so far, but we still have some problems. And as you can see here, many molecules, we can predict the properties of these molecules with a very low error in comparison to the literature. However, we still have some problems here. Some molecules are still jeopardizing our results. We still need to scrutinize these results in order to check what are these molecules, why they are somehow jeopardizing the accuracy of our predictor, and so on. This work, we have a first draft. We're about to finish this work, and we hope to publish it as soon as possible. The second topic is graph-based clustering. When we talk about graph-based clustering, it means we are taking molecules, samples from our data set, and representing those samples into a graph. It means each molecule will be represented by a single vertex in this network. And the links in this network are built according to the similarity between molecules that are represented by those nodes. It means if you have two nodes and these nodes represents two similar molecules, they are connected. What is interesting here is that we can analyze the structure of this graph and get very important information. For instance, in contrast to k-means approach for data clustering, we can highlight clusters with very complex shapes. On the other hand, k-means can only highlight Gaussian-like based structures of the clusters. So with this graph structure, what do you want to analyze? We have three main questions. First one, are the samples in the QM9 organized into clusters? If so, we can perform better analysis of this data set. For example, by analyzing the clusters, we can analyze what's the feature represented in these clusters, try to establish a connection between the features and the properties represented in these clusters, trying to observe if is there some cluster that provides that anomalous molecules that introduce higher error in the prediction, and so on. By our results so far, we confirm that this data set is organized into clusters. The second question is, can topological features of the samples be used to characterize it? It means, by analyzing just the relational structure of the graph, take a node and grab some measures of this node. Can I give some information about the molecule that it represents? For example, this node represents a molecule with high energy or lower energy, and so on. We still didn't get good results in this to answer this question so far. This is still undergoing research. And finally, highlighting this problem that I have here in the Gabriel's work, by analyzing the graph structure, can I isolate those molecules that introduce this higher error using these topological features of this network? Here is a coarse-grained version of the cluster structure of the QM9, in which circles represent, each circle represents a cluster. The size of the circle represents the number, indicates the number of molecules grouped together. It means larger circles will be larger clusters. And the connections between these circles represent that there are molecules in both clusters that are similar to each other. So you can see that we have many clusters with large size, which means clusters that hold most of the molecules or a large number of molecules, but we also have many small clusters that can be analyzed and can be isolated from the data set. These are still undergoing research. The third one, it's which I call biased clustering. Instead of performing orthodox cluster, which is unsupervised learning, here we are mixing clustering algorithm and optimization algorithm. What we want to do with this kind of supervised clustering, instead of providing the clusters using just the features of the data set, we want to get some informations that I have besides the features. For example, a property of a molecule. I know that some molecules have high energy or lower energy, and so on. And I want to use those external information to modify the shape of the cluster. It means, given a set of features, I want the systems to modify the weights I'm giving to each of these features in order to maximize or minimize an target function. For example, if I want to produce clusters that minimize the energy variance inside each group, I perform the data clustering using the k-means. Then I run the optimize algorithm to set up new weights for each feature. Here we have an illustration. I have this internal circle in the first step, in which all the features have the same weights. As the system evolves, some features get higher weights, some features are almost ignored in order to achieve this kind of objective. I mean, minimize or maximize some property, desired property. Regarding our collaboration with Professor Juarez, I can point three main points, three main lines. The first one is a correlation analysis, in which we run some correlations with local data. From this analysis, we have two papers published, already published. Paper one is a paper of Paulo. Where's Paulo? And also a paper from Quiles. Both use the same sort of correlation analysis, but for the paper, Quiles paper, we also used a machine learning approach to perform prototype selection. We use here a k-means approach, k-means algorithm, to select one among several molecules. For example, if I have 1,000 molecules in my data set, and I want to select 10 molecules that represent this 1,000 molecules, the most representative molecules inside this quadrant, a subset of this set. So we use this algorithm to select this subset of representative samples. And also, we also wrote a code, an algorithm for molecular absorption. This work is still, it's not published yet. Chris is finishing the writing of this paper, and we hope to submit this paper soon. What this algorithm is for example, given a cluster of 13 molecules of silver, 13 atoms of silver, we give this cluster and also a molecule, in this case CO, 6 CO molecules, as input to the algorithm, and the algorithm returns the position of each of these CO molecules, where this molecule should be absorbed. As I mentioned, this paper is not published yet. So, just revisiting our mission, I believe from a perspective that our mission here, the first is to help problems that we haven't seen with machine learning methods, and as a consequence, to provide new developments and improve methods from machine learning, from the machine learning perspective. But to do this kind of research, we need two things. First, we need data and collaborations. I cannot do applied machine learning without the collaboration with the applied field, the applied area. I cannot, inside my group, develop machine learning methods to material science or to chemistry without this sort of collaboration. So here I invite you to talk with me later, and maybe we can establish some other tasks in which my group can help your group to solve some local problems. Here's my team. I have two undergrad students. Felipe Calderan is going to present a poster in a couple of minutes. Bruno Raceira is starting his scientific initialization in the next month. And two PhD students, Gabriel Pinheiro, who is also going to present his paper, and André Oliveira, who is starting his PhD also next month. Actually, Gabriel is a master's student. He's going to defend his master thesis next month and also start his PhD under my supervision. So I still have room for some tasks here. So collaboration is welcome. I would like to acknowledge FAPESP CAPS CNPq and Shell, also Professor Eudes Filet, Juarez, and Professor Ronaldo for the preparation along these developments. My students, Gabriel and Felipe, especially who are with me for a while, and QT Nano students, especially Jonathan, who helped us with our internal developments, and also Jacqueline, who gave us all the support we need in this project. That's it. Thank you very much.
Okay, so we are now open for questions.
I have basically 3 questions. In the case of the graphics there, okay? Yes, that's right. The KM9 set is composed by a number of 130,000 molecules spread from different sizes.
Yeah.
They have from number 3 up to 29 atoms. Then I expect if you try to group them, they will group basically well because they are different size of molecules.
Not exactly.
Okay, exactly. Come on, that's another point. And then the number of molecules are different from the different sizes of molecules. And for some particular size, I think I'm not so sure, I think 28 or 27, they have 6,000 isomers in one of the sets, if they are molecules with the same size. My question is for the KM9 set is the best set to test the graphical-based cluster analysis.
I cannot answer this question. I need to have all the data sets in order to compare those data sets. The only thing I can assure you is that the QM9 can be split into several groups regarding the properties or the features I'm analyzing to perform the data clustering. If I take a set of features, I have one clustering structure. If I had another set of features, I have another clustering structure.
Yes, because why I make this comment? Because the set itself is already clusterized easily based on the number of atoms, the molecules.
Yeah.
But not clusterized based on the number of properties.
Number of properties and
The type of properties. For example, if you look at the homolume, how the spread of the homolume, how the system is connected.
Okay, but let me clear something. To perform the data cluster, I'm not using the properties. I'm using the features.
Yes, but features means only the structure.
Yeah.
Yeah, this is the point.
I take these miles of each molecule, extract the features, and based on the similarity of these features from several molecules, I perform the data clustering.
Yes, exactly. But if you go beyond, that's the point. Okay, the second question is concerned the data that Gabriel is doing. We have these large arrows. My question is there some way that you could understand why some particular molecules give this particular arrow on the cell, on the data, on the machine learning training? For example, that particular cases.
We don't know yet. I cannot isolate those molecules based on their features. I was expecting to perform this clustering analysis and from this graph structure try to grasp where are these molecules positioned in order to identify them, but we still didn't get this.
Okay, but at the same time, we are able to identify who is the molecules that give the large arrow, and from the chemist perspective, we are able to see how those molecules, how much they are different compared to other ones.
Of course.
From the chemist point of view, maybe we should combine the chemist perspective with the machine learning perspective to try to identify and explain it.
From the machine learning perspective, I cannot give you this answer.
But from
chemistry, from the chemistry perspective, yes.
KM9 is to spread over the type of molecules, in the sense that it will depend on which kind of filter you will employ in the filterization process. In the way of the smiles, for instance, smiles look to the connectivity and the type of atoms and so on, but don't look, for instance, for the angles. So if you have a molecule that have a completely exchange angle, it will be very a part of the rest of the data. So you will have like 2 molecules in a very isolated dot in the graph, and they behave very different from the others. So if you try to look at a machine learning that fit all the data, it will commit large errors for these small isolated points. If you employ a different data set with very similar molecules, so it will not, this kind of errors will not happen.
My last question is the following. For example, yesterday, Professor Shu, he provided, I think, this statement that's concerned for example, we could use machine learning to search the PDF files of a papers that is published in the literature instead of to input up all the data ourselves. For example, use your expertise that you have, or you and Ronald, for example, could be able us to have a tool if you could run over like 10,000 PDF files from a particular field, for example, perovskites. We run over the 10,000 papers from perovskites and finds out there what is the trend that's going on concerning the experimental data.
Yes, we can, but it's not my expertise. It's like, I've been working with PLN, natural language processing, but in the context of social data, social network analysis. I've never worked in this kind of specific in the sort of work. I don't know if Ronaldo have done some.
It may be possible, but the answer is not that easy because the methods are cannot really free text right now, but we can try to find some patterns in those articles and try to, yeah, it could be an interesting idea. And regarding your second question, Luis, my master student who is there, is working in procedure to try to understand from the machine learning perspective why some models have higher errors. So we invite you guys to talk with him in the process.
Okay, then. So let's thank Professor again.